All right, it is now my great pleasure to give it over to Dan. He is going to moderate the last hour of this 24-hour event. And I will just give it over to Dan. Bye.
All right. Okay, let me share my screen here. Can you see my screen?
Yep, I can.
Okay, let me welcome everyone to the final hour of our 24-hour event, appropriately enough on the future. My name is Dan Qaurooni. I'm an anthropologist of technology and the moderator of this panel. And over the next hour, we'll delve into the transformative impact of AI across various sectors of society. And our esteemed panelists are on the forefront of these transformations. So I'm certain that this will be a rich set of presentations and discussions. Okay. So here's an outline of the hour. I'll briefly introduce the topic and focus a little bit on ChatGPT as a sort of example of the recent trends in machine learning and AI and its implications for different sectors of society. Each panelist will then spend between 5 to 10 minutes, 10 minutes max, to introduce themselves and present. That will leave us enough room for a discussion towards the end, and then Katy, our host and last panelist, will deliver the event's closing remarks. Okay, so introduction, yes. As I'm sure all of you know, AI has been around since the middle of the last century, at least, so for about 70 years, but there are signs that something might be shifting in the relationship between AI and society. And over the last couple of years, this relationship has experienced some unprecedented changes. AI has become a subject of unprecedented attention, interest, and anxiety. And so the poster child for this trend is of course, ChatGPT, OpenAI's world-famous chatbot. And so by way of introduction, I will focus briefly on GPT. Starting from the T. The T in GPT is for transformer, which is a particular neural network architecture that I don't fully understand and I'm still learning about. But suffice it to say that it's the main technical innovation behind ChatGPT. And transformers became famous with this now classic paper published by researchers at Google and the University of Toronto in 2017, called "Attention is All You Need." This paper became an instant classic. Its citation count is around 100,000, 6 years into its publication, which is kind of amazing. And as a funny aside, the paper's title has inspired a whole subgenre of paper and publication titles. But yeah, the T in GPT stands for transformer, which is this neural network architecture. The P is for pre-trained, which is refers to the fact that one of the bottlenecks in machine learning had been sequential processing, and transformers had introduced a efficient parallel architecture that offered a solution to this bottleneck. And as a result of this, you can achieve massive scaling effects in your pre-training and conduct pre-training on tons and tons of data. GPT-2, GPT-4's numbers, I couldn't find them. I don't believe they've been released, but GPT-2 as an example was trained on 45 terabytes of internet data. And finally, the G is for generative. Models like ChatGPT are basically token predictors, which means they're a very complex version of the modest autocomplete that you're familiar with. And the cognitive psychologist Alison Gopnik summarizes the way these token predictors work for the layperson in a way that I find very helpful. It just says a model see the first part of a text that someone has written and then try to predict which words are likely to come next. If a powerful computer does this billions of times with billions of texts generated by millions of people, the system can eventually produce a grammatical and plausible continuation to a new prompt or a question. So they're basically completing the prompt that you give them. So what's been the implication and the impact of ChatGPT's release, which I'm sure everyone is at least somewhat familiar with, but let's go over these in a couple of main areas, which I find interesting. So as these headlines show, the cognitive capabilities of GPT and large language models in general have triggered a lot of debates. The headlines that you see here reflect a sort of debate, an ongoing debate about cognition and consciousness. It's become a very polarizing topic. There are key figures like Douglas Hofstadter and Noam Chomsky who are expressing sort of skepticism without denying the advanced functionality of these systems, nevertheless expressing skepticism that these machines do anything but mimic the original, so to speak, when it comes to advanced human functions like language or symbolic learning or intelligence or consciousness. And then conversely, there are stories like the Google engineer story, and people claiming that these systems are sentient or they're on the verge of achieving AGI, artificial general intelligence. In science, the impact on the scientific community has been complex as well. AI has been used to solve protein folding problems, which shows its potential for scientific advancement, but there are concerns about its reliability and the implications for universities that face challenges about GPT and similar systems use in terms of academic integrity. It has fooled scientists and attorneys and generated with fake abstracts. People have talked about the risks of science denial. In education, the headlines similarly paint a very complex picture. GPT-4 has passed professional exams. And this might sort of signal a potential shift in how AI tools could supplement or transform education. Maybe traditional academic practices like the college essay are now obsolete. Should these tools be banned or integrated into educational settings and questions of this sort? In labor, another very controversial area, GPT can be utilized to automate tedious aspects of jobs. I know I do that sometimes, and potentially increase efficiency. Yet, like many advanced technologies, efficiency is only sort of the tip of an iceberg that can be exploitative in ways that remain invisible. So GPT has reintroduced the ethically sort of charged topic of unseen human labor in relation to AI development. And finally, in politics, more broadly, prominent scientists are sounding the alarm about the potentially dangerous impact of AI, sort of the critical need for careful management and awareness of AI's impact. Europe has moved to regulate AI with potential new laws. President Biden has issued an executive order to jumpstart the creation of safeguards for AI. And AI is being compared to the atom bomb and OpenAI to a sort of corporate version of the Manhattan Project. So the analogy highlights perhaps the sort of the perceived magnitude and high stakes of AI's impact on society. Yeah, so there's much to discuss here, but I'm going to now turn it over to Dr. Soumya Ghose. Dr. Ghose has been involved in developing machine learning, segmentation, and registration methods applied to oncology, healthcare analytics, and cardiology. He's the principal scientist at GE Research Healthcare. Dr. Ghose, are you going to present the slides yourselves, or would you like me to?
Dan, is it okay if I share my screen and present my
Yes,
slides?
yeah, sure.
Okay, let me do that.
And there's going to be, I'm sorry to interrupt, one reminder that there's going to be a sort of 10-minute timer in the interest of keeping the discussion focused. If you finish earlier, then we'll have more time for questions. Thank you.
Yeah. So thank you, Dan. Thanks for the introduction, and thank you, Katy, for the invitation over here. So we are involved in a human biomolecular atlas program with NIH. The primary objective is this program is to map cells and create a 3D map of different body parts and organoids and organs at a single cell resolution from a molecular perspective and from a single cell perspective. Now, there are different centers involved in this process. This is funded by NIH. This is going on for some years now. And there are like tissue mapping centers which collects the data, and there is a hive which coordinates among each other to create those 3D maps over here. So a number of organs have already been mapped, mostly at this point of time, mostly 2D, and the 3D process has just started over the years from now. So we are in the tissue mapping center, which involves with 3D reconstruction of the skin samples or the skin tissues over here. So this is our group, a multidisciplinary group consisting of biologists, molecular biologists, microsystem engineers, and pathologists from University of Pittsburgh and GE Healthcare Research. So this is how we address the problem. So after 3D collection of the samples, which are basically the biopsy samples, certain biomarkers are selected. Primarily, we target mutation biomarkers and like immune cell biomarkers, also with blood vessels, because that is very important for us too. So once that has been validated, those antibodies has been validated, what we do is a multiplex imaging system, where in each round a particular biomarker is selected, and that is used for dyeing that particular skin samples. So for each volume, we also create a CT map of that, just to get an idea of the volume, how it looks like before we slice it up. Now it has been sliced into 26 serial sections, and each serial section goes to a round of around 20 biomarkers, as shown over here. So once that has been created, the next step is basically segmentation, and then the reconstruction happens. And once we can do that in 3D, then we can start asking all biological questions in 3D, which is a big jump in terms of what we used to do before. So we have already done this for around 40 patients with different diverse backgrounds and skin colors, which is also very important for us. So just to have an idea about how those different biomarkers looks like. So this is a hair follicle that you can see, and you can also see the different biomarkers that has been collected for this particular samples. We are primarily interested in the immune biomarkers, the blood vessels biomarkers, and the different mutations that may happen due to UV exposure of the skin. So the first step for 3D reconstruction is segmentation of the cells and the biomarkers. So the way we do it, there is like two parallel models going on in this case. So in one of the models, we use a multi-channel deep learning model for segmenting of the cells. There is another model which is unsupervised, which segments of the biomarkers and separates the foreground and the background. The advantage of this process is you can highly parallelize this system. So the cell segmentation and the biomarker segmentation are parallel to each other. So what happens is you can also start combining different biomarkers together. For example, we were interested in the p53 mutations going on in the epidermis. So we started combining the probability maps of p53 with AE1, which highlights the epidermis, and start combining them to get the p53 mutations going on in the epidermis. And then you can overlay with the cell segmentation and find out the cells which shows those kind of mutations. So one advantage of this model is no manual annotations are required for the different biomarkers. It's highly scalable, so you can. We have already tested this system with other tissue types, for example, kidney and brain tumors, and it works fine. And it also helps us to explain and understand biologically important questions, like, for example, like what are the p53 mutations in the epidermis. Now, once those biomarkers has been segmented, the next step is 3D reconstruction. Now for 3D reconstruction, we have the serial section. So it's like creating a 3D volume from those stacks. The way we do it is basically we select a reference slide somewhere in between those 26 serial section based on the image quality. We quickly do an affine registration based on patch-based correlation between the serial sections that happens. And then that is followed up with a B-spline-based registration, which is deformable registration, which then can take care of the deformation that may happen during the acquisition process. So now once that happens, what you get is a bunch of transformation maps for each of those serial sections. And that transformation maps help us to transform all the biomarkers into a 3D map of the skin samples of the biomarkers that we're interested in. So now what we can do is basically overlay the biomarkers over the skin samples as shown in the picture over here. You can also generate a 3D mesh of all those biomarkers as you can see over here. So all the cells are in 2D and 3D are indexed. We have the cell centroids, shape and appearance feature. We can create the 3D maps, and then we can start asking important biological questions. So this is how the overlay looks like from one slice to the other. And this is how the mesh map looks like from one slice to the other. So you can see everything in 3D. And then once you can have that, then you can start asking the questions that you're interested in. For example, we were interested in what is the distance of the blood vessels from the different immune cells, which is a kind of a good feature set for understanding how the cancer is evolving. So Ingnan did this work with Katy, and this is how the map looks like. So you can think of it as like a feature set, the different immune markers corresponding to the blood vessels, and let's say what is the distance from their different p53 mutations, which can then be used in a machine learning framework to understand the progression of a disease. Similarly, you can start looking at the different clusters of the immune biomarkers that are getting created with respect to each other. As you can see, we are observing the T helper and the T killer cells over here. This is a very interactive system that has been developed by Ingnan, and it's available online. I'm happy to share those links after the presentation. What we also understood is like distance in 3D is always less to the blood vessels of the different cells compared to distance in 2D, which is quite understandable. The other important thing about looking in 3D is like, this is how the 2D map looks like. As you can see, it's very sparse for the different immune markers. But as soon as you start constructing the 3D, you can quickly see the relevance of 3D and the clusters of the biomarkers that are getting created in 3D. So more number of cells could be seen in 3D, more number of clusters are seen in 3D, which is quite relevant in terms of biological context. So I can probably stop here, and maybe we can have the questions later on.
Okay, thank you very much. Feel free to post questions in the chat, and I'll collect those, and we can have a discussion after everyone has presented. Okay. Okay, our next speaker is Dr. Y.Y. Ahn. Dr. Ahn's a professor of informatics at Indiana University, and his research focuses on developing network science and machine learning methods and applying them to complex social and biological systems. Would you like me to, do you want to share your screen, or?
Yeah, I'll share my screen. Yeah.
Okay, go ahead, please.
Yeah, can you see my screen?
Yes, perfect.
I think you want to reset the timer. Yeah, hi, my name is Y.Y. Ahn from Indiana University, and I'd like to talk about some black hole and space of knowledge. So today I want to kind of pitch kind of new opportunities for better macroscope by using the representation learning that's at the heart of this deep learning and AI systems. Yeah, first of all, I would like to mention that observation is the bedrock of science. Like being able to observe and measure is like really the important foundation. And the instrument really matters, right? Whenever we have a new telescope in the space, we get a slurry of new innovations and results and advancement in science. And if you look at kind of scientific enterprise itself, there are lots of data and measures and maps that just coming online. And that's kind of a lot of opportunities and data, and it's kind of new possibility for new measurement. And there have been a lot of maps, very cool maps, including Katy's, that kind of maps out how the science is related to each field of science is related to each other and where does the scientific activity is happening. But my question is like, what could be kind of the next step beyond these network maps? And I feel like the map shouldn't be just a visualization and just shouldn't be a map. And the map to go to the next step, it should allow like really meaningful measurement that is interpretable and meaningful and quantitative. And again, I would like to pitch the idea that the representation learning can be a really nice way to create a new macroscope, especially using this compressed representation that these machine learning systems produce. And a cool thing about this AI system is that this neural network learns the meaningful and interpretable space. For instance, this is from word-to-back method, a precursor of all this LLM method. And people found that it is possible to find semantic axis as a kind of geometric alignment or axis in this space. And that allow us to interpret the space nicely. So if you think about this kind of a concept like space of words, space of material and images and so on, and that allow kind of that pre our imagination in a way. So by thinking about, oh, what if we conceptualize the space of words where words move over time? And that kind of easily lead to this type of questions like, okay, how does word move over time? And in this paper, they show that, oh, you can actually trace how words meaning, the semantics change over time with respect to other words. And that's in part possible because now we are thinking about the space of words, which is really kind of a visceral and natural to think about for us. So I've been working on so-called science genome project where we ask the question of like, can we imagine the space of knowledge where we can do measurement, quantitative and interpretable measurement. And by having this map, can we model the whole scientific enterprise just as if we model the murmuration of birds or foraging of animals. And also you can think about like where's the black hole in the space of knowledge, where people get attracted to, but it's hard to see from outside. So here are some results I want to share. First, the study we did was kind of embedding the journals, scientific journals, based on the citation network. And in doing so, we can kind of do fun measurement. Like for instance, can we kind of recover this XKCD comic where we align everything, every journal from like hard science to soft science, or like social to biological sciences. And it's possible the space kind of captured that semantics. And furthermore, we can even do analogies. We can now, because it's a space, vector space, we can add and subtract vectors. For instance, KDD and ICWSM, they are famous like computational social science conferences. If you keep adding machine learning and subtracting sociology, we get more and more technical computer science conferences that focus on machine learning. While if you add sociology and remove machine learning, we get more social science journals. And this can be done across the whole space. You can start from like Journal of Climate and keep adding kind of a social science, and it eventually become like weather forecast. And then by adding physics, you get like climate dynamics. And you can do this for individual papers. This is kind of the map where we put individual paper based on their title and abstract into embedding space. And you can kind of see nice structure with the empty space in between. And once you have this map, you can place on top all kinds of information on top of it. So this is, for instance, the number of papers across the space. This is where kind of the most disruptive science is happening, defined by the disruption index measure. And you can also compare like how different countries produce different types of science. But then like the question remains, can we really interpret this space as if it's a real space where we can do measurement? And the hint was from this study, which is in press and PNAS, where we asked the question of like, can we map the movement of scientists and then using that to map the institutions? So one surprising thing was that if we apply this really basic method called word-to-back on the trajectory of scientists, then you recover the space where the distance between institutions kind of obey the gravity law of human mobility. And that means if there are more people moving between two institutions, they get pulled in. And if there are fewer people move between institutions, they get kind of not pulled in as strongly. And as a result, we showed mathematically that they are equivalent to the gravity law of human mobility. And this gives us kind of high resolution map of all the institutions. And interesting thing is like we can look at an axis from like prestige institutions to low prestige institutions. And then if we project every institution on that axis, it highly correlates with the actual ranking of the institutions, for instance. So this captures a lot of interesting semantic axis. And this idea can be combined with this embedding idea. I'll skip this. And the idea is we can have a trajectory of scientists in terms of paper titles. And then we can use those title sequence to fine-tune text embedding model like BERT so that the text embedding satisfy the gravity law of mobility. So this produces a conceptual space where we can put any sentences. And then the movement between two topics should obey the gravity law. And as a result, we get this embedding space. And that can be interpreted as kind of literally with the Newton's law of gravity. For instance, we can calculate the gravity potential across the space. And this is a kind of space of biological research. And then an interesting thing in this landscape is that to go from cell-based biological research, like more fundamental molecular biology, to human biology, there's no direct pathway, but you kind of should go through the animal research. And that capture how people move in the space of biological research. And it also allows us to predict whether two scientists will collaborate together in the future. And it opens up way to model the whole scientific enterprise using very physical models. Yeah, so yeah, at the end of the day, we found some ways to map the space so that the space can follow the law of gravity, and that allow us to do concrete interpretation of the space. And I would like to thank my team and thank you for listening.
All right, thank you very much.
Yeah.
Our
thank you.
next speaker is Dr. Abe. Dr. Abe is distinguished research staff member at IBM Research AI. Yes. Manager of Foundations of Computational and Statistical Learning Group within the Foundations of Trusted AI Department of IBM Research. Would you like to share your screen or?
No, if you can share, that would be great.
Okay, sure.
I'll just ask you to go to the next slide. Thanks. Yeah, thanks for the introduction. I think some of the title actually was a little bit outdated. There's been there have been some changes in the last couple of years, but anyway.
Good. Happy to talk more.
Yeah.
Okay.
Yeah, so, well, thank you very much. It's a great honor to be part of this great event and panel. There have been some very exciting presentations, and I'm afraid mine will not be as visually enticing as the previous presentations, but I'd like to present some perspective, I guess, coming from the industry, some perspectives on the challenges that we may still face, within AI. Oh, you're putting it into the slideshow.
Sorry. Yeah. Let me just start the timer, which I forgot to do. Please go ahead.
I see. Yeah. The previous slide, please. Yes. Right. Oops. Right. Yeah, so as you know, everyone knows, and as articulated by Dan and everyone, generative AI, particularly large language models, has made very impressive progress in recent years, and so one wonders what challenges remain, and there are a couple of areas that I would like to call out, and one is the trust and governance. That the trustworthiness of AI has been an important agenda for a number of years now with the surge of deep learning methods and applications. But I think especially now in the context of foundation models and large language models and their ubiquitous, presence in our daily life, this is getting even more pressing. And there are multiple dimensions within this trust and governance area, including accountability, things like factual correctness and whether the certain things that come out of the model, whether you can track them to the sources, all these things. Explainability is also very important. If the model predicts something, you would like to know, why the model is predicting, what it's predicting, and whether it's justifiable and so forth. There's the fairness issue. If the data are biased, the model's judgment tends to be biased as well. And, how can you correct for it and so forth? There's a serious issue on privacy. It's also related to accountability and traceability, but, who owns the data? Who owns the knowledge that comes from the data? How much say do you have on the privacy of your data and so forth? So this is a huge area of concern and ongoing research. The other dimension that I would like to call out is the question of modalities. So these foundation models that have been trained using billions of data, they have been empirically proven effective. But I think it's fair to say that they have been proven very effective in modalities that are sort of specific to human cognition, especially images and texts. I saw some very interesting work by the previous panelists on biological data and so forth. And yes, there's a lot of exciting work there, but there are questions to what extent these foundation models can work well in domains and applications other than cognitive tasks. And that may include numerical time series data, biological data, geospatial data, event stream data, and so forth. So these are two big areas, I think, where challenges lie. If you can go to the next slide, please. So this is kind of a slide that's depicting some aspects of accountability and explainability, and sort of a specific viewpoint that I'm personally trying to promote. And that is essentially trying to leverage logical and causal frameworks to bring accountability and explainability to these huge foundation models. So for accountability, there's this known issue of hallucination, and people realize that in some way you need to incorporate the domain knowledge and the learned knowledge together in a coherent way. And for this, logical consistency is key. For explainability, there are many aspects of explainability, and we've just seen some very, very interesting visual explanation afforded by leveraging the embeddings and so forth. I think that's very interesting. But in many business and industrial scenarios, explanation needs to justify whatever actions you may be recommending. And in order for you to be able to do that, you do need to have causal coherence, in the sense that your model needs to be able to predict meaningfully in a causal manner. When you do something, you need to be able to predict the effect of it. So that's an important aspect of explainability. And what I'm sort of trying to depict here is that through bringing the frameworks of causal modeling and learnable logic on top of these foundation models, I'm hoping that we can enhance these aspects of foundation models. If you can go to the next slide, please. And on the question of modalities, as I mentioned, many important AI applications involve data modalities other than those that are common for human cognition tasks, like language and vision. And I think there is relatively less evidence and reason to believe that these neural network-based methods that are inspired by the brain can work better than other type of competing approaches. And some example application domains include climate and environment data and applications using them, life science and health data, enterprise and IT data. And it includes some of these different kinds of modalities. And if you can go to the next slide, please. So there are here some examples of such data modalities in these application domains, and these are from projects that I have personally been involved in. I was involved in a bio fuel or agriculture project with Purdue University, right from Indiana, and for that sort of application, you collect a lot of data from drones and so forth, and including things like hyperspectral data that you need to analyze. And there's another digital agriculture project that I was involved in with UIUC, and for that, you need to deal with things like crop genomic data. At the bottom is an AI for IT project that I'm still currently involved in at IBM, and for this kind of application, you need to analyze event stream data. And for these types of modalities, there isn't yet a foundation model that you can leverage that's been trained with millions and billions of data. And so going forward, I think asking the question and exploring whether this type of general big models could be made to work well in these domains, I think is a very important and interesting problem to pursue. So let me stop here. That's what I had to say. Thank you.
Thank you very much. Thanks for great presentation. And finally, our last speaker, besides being our host, is Dr. Katy Börner, Professor of Intelligent Systems Engineering at IU. Katy specializes in scientometrics, infometrics, science of science studies, network science, and cyber infrastructure. Okay.
So you might have realized that this panel brings together really the mapping of science and the mapping of the human body to atlasing efforts that are very near and dear to my heart. And I think there are interesting synergies between the two, because if you start mapping the human body at single cell resolution, and you can go to the next slide then you will need to empower and engage thousands of experts around the globe to work together and to agree on gene names and cell type names and even anatomical structure names. You would think that is agreed upon, but no, not necessarily so. And you will need to create massive knowledge graphs and ultimately need to visualize them so that human brains can see patterns and trends and outliers. And of course, you will need to bring in machine learning in all kinds of forms and shapes to do segmentation, 3D alignment of tissue sections, as Soumya presented here. And you will have to do this across scales from the whole human body and probably beyond, because we are social creatures. And so we live in social environments that need to be also ultimately covered. But here, let's focus just on one human body, male or female, and then zoom into an organ, here the kidney, or a functional tissue unit. You each have, if everything went well, if you're not born early, then you have about 1 million renal corpuscles in each of your kidneys, so 2 million total. And because of that redundancy, you can actually lose a kidney or give it to another person who needs one, and it will still work. So we are trying to understand how these functional tissue units really work with the structures they have. And then you would zoom in to see podocytes here in this example, and zoom in even more into the nanoscale level to see genes and proteins and lipids and metabolites. And so to, again, make that work and speed up the construction of such a human reference atlas, we have been trying to bring together people that have lots of expertise in federating data sets and analyzing and modeling them and visualizing them with the experimentalists, which generate some of the most beautiful data about our healthy human bodies. And there are different 24-hour events. We hosted one last year on the Human Reference Atlas. So you can go check out who presented there and what they presented. We will have a similar event next year, I believe. And we are now also trying to see if we can use the vasculature as a common coordinate framework, as a latitude-longitude system across the human body and across scale. So on the next slide, you see a paper that describes how many different consortia, we now have 17 consortia, are working on which organs, and ultimately agreeing on a data structure where you have anatomical structure pathonomies. This is the image in the lower right, linked to the cell types that are commonly found and located in those anatomical structures. And then, of course, also linked via bimodal network to biomarkers that are typically used to characterize these cell types and cell states also. And it's a non-trivial community consensus finding effort that we are doing here, because again, many have to agree on this framework of representing this human body in this way and agree on the metadata standards ultimately also. So on the next slide, you see Kaggle competitions which we have been running on segmenting functional tissue units across different organs, here 5, on the top right, and also segmenting blood vasculature. And initially, that was all done in 2D because that's what most of the data is like these days. But more and more 3D data is coming in. And so currently we are hosting, together with the HOA team led by Peter Lee at UCL, a Kaggle competition that looks at vasculature segmentation in 3D. And we are super excited to see many teams participating. And there's a link to it in the lower right. And there's quite a bit of prize money associated. So please feel free to point your students and others to it. On the next slide, you see the promise, some of the promises of AI. So could text-to-image-based models and ultimately also image-to-text-based models be used to speed up the construction of such an atlas, making sure that more people live healthier, productive lives, live longer and happier, and if disease moves in, can be cured and in a meaningful way so that there is healthy life after that. And so I have been exploring all kinds of different tools, and team members also across HubMap and the other consortia as well, trying to really understand what is possible when and what data sets, highly curated data sets, are the best input to training these models. And you see here on the left a DALL-E rendering of organs connected via vasculature about 6 months ago, and then we recently did another trial on this. And of course, it's totally wrong because the input data is not highly curated data. So I think we will very soon see models that are trained using highest curated data, and it will look very different. And I think it really requires this combination of human and machine intelligence to create this human body map. On the next slide, I wanted to leave you with an outlook to the future, where I believe we will have to find ways to live in harmony and symbiosis with AI. They are our children, just like some of us have teenagers, and they are not always nice. They're sometimes rather prickly if you have strong kids, which you grew up in your household. And ultimately, however, you want them to succeed and you want them to give back, but also do their own things. And so it will be very, very interesting to live in a future where both of these are possible and where we live very, very closely with AI. So here you see Luddy Hall. We have Amatria Heng in our environment. She is a sentient architecture that we welcome in our lives, and that has inspired really many, many artists to come into the engineering department at IU, but also inspired many, many creative endeavors. And so hopefully we have a symbiosis and not wars raging. And hopefully also AI can help us live healthier lives and understand science a little bit better. The endeavor we all do together. And I will give it back to Dan.
Okay, thank you very much, Katy. All right, we have a couple of minutes for discussion. If there are questions, I can field those in the chat, or comments on the presentations by other panelists. Feel free to jump in. Anything? Okay.
I had a question to Soumya. Maybe it's a little technical, but when he was showing 2D and 3D maps, sorry, I guess maybe I missed what you said. But what were the axes of those 2D and 3D maps or spaces?
So these are all in Euclidean space, I would say. So what we do is basically we have a volume of, let's say, skin samples that is sliced. We take a CT of that before we slice them, because you need to slice them to get all those biomarkers for individual slices to have a dense reconstruction of those. But we have that CT map to which they all get registered eventually. So I would say like it's an Euclidean system of all those biomarkers that are getting reconstructed into. that city space.
Okay. City space, you said?
So yeah, so what we do is basically we are from GE Healthcare, right? So we have those machines like CT machines, MRI machines. So before we do that slicing of those individual 2D slices, we take a 3D map of that in CT. So now once you have done that, now we can register them back to that CT
so those
space,
are more,
I would say.
okay. So those are more, faithful to the actual 3D
That is correct, yes.
structure, as opposed to the embedding space that Yeol was describing. That is sort of in the representational space. So those are very contrasting types of spaces.
I mean, it's very common to use an embedding space for those biomarkers too. So you can have a UMAP kind of representation where all those biomarkers getting clustered into different clusters. You can do that too. And it's very common for, especially for single cell RNA-seq, where you don't have the spatial location of those biomarkers. But for this particular project, we have those spatial locations in 3D for all those biomarkers.
I see.
So you can do both. You can do UMAP kind of mapping or 3D mapping. So, but for biological phenomena, 3D mapping makes more sense, like the distance from the blood vessels, the distance from skin surface, let's say. Those are like features that you would like to track in 3D.
I see. Thank you. And I also found Yeol's work, of having these spatial representations in the embedding space and constraining them, with the gravity structure. I thought that was fascinating. And we're also exploring ways to constrain the embedding space in some different ways. And as I mentioned, some of that could involve the notion of causal coherency or logical consistency or something to constrain the learning of the models. But that's still work in the future, really.
So
Okay.
we all know, like, a question. So we all know, like, it's really important to understand that modality that we are dealing with for any particular embedding or restricting those causality. So I would like to understand how different people are using that. Like, we have so much diverse domain over here. So how do you plan to, for example, for GPT hallucination, how do you stop that for any particular domain?
Yeah, so what my personal perspective was to constrain it with, let's say, logical reasoning, and there could be multiple ways to try to do that. But I mean, in the abstract, you might do that through some sort of feedback loop, like in how do you call it, RFAIF, I guess, reinforcement learning with AI feedback. But with that sort of consideration, this, again, it's really speculative future work. It hasn't been. I think people are trying or starting to try, but yeah, it's very much work in progress, I think.
Thank you.
And in fact, in the Human Reference Atlas Working Group this week, Katy has more information on this, but somebody was presenting on OntoGPT, which was a combination of GPT and ontologies. Katy, their name escapes me. Do you remember who it was?
Yeah, Chris Mungle. He is.
Oh, yeah, yes. Yes, basically making the same argument that that the shortcomings of these, including hallucinations, these transformer models can be basically addressed by carefully constructed and curated ontologies.
In fact, that brings us back to the point about spaces and what kinds of spaces we're dealing with and how we conceptualize them and how everything rests on those conceptualizations. 12 months ago, so for the last 10, 15 years, the world, so to speak, thought about knowledge exclusively almost as networks. knowledge graphs, ontologies. When people like me started to talk about embeddings and feature space, they were looked at strangely and looked at our artifacts. They were looking at my maps and said, what ontologies were you using? Because they made so much sense. And I said, we don't use an ontology at all. It comes about through embeddings making sense of themselves through a neural network approach. Now the pendulum has swung over to the continuous space of feature space, and the goal is now to link the two. The world of the concrete, of concepts and concrete objects whose existence has been asserted or affirmed, and the world of what could be, the world of probability.
Okay, Katy, I think we're almost out of time. If you...
What a great last word, Andre.